Welcome, everybody. Welcome to episode 3 of AFDO's Cannabis, Hemp, and Natural Psychedelics Committee webinar series. My name is Thuy Vu, and I'm one of the co-chairs for the committee. And before we get started, I just wanted to say that the views and opinions expressed during this virtual event are those of the presenters and not necessarily the Association of Food and Drug Officials. Participation in the virtual event does not necessarily constitute an endorsement of the speaker's views, products, or services, and this virtual event cannot be used to train AI models. So I wanted to welcome back Dr. John McKay for his part 2 of a part 3 series. Dr. John McKay is recognized as a recognized educator and scientist, and the founder and CEO of Synergistic Technologies Research LLC. Dr. McKay has held leadership roles at the Newbridge Global Ventures and Waters Corporation. He also contributes to scientific literature as a journalist and editor for Extraction Magazine and Terpenes and Testing Magazine, and serves as an assistant professor at the Robert Lerner, MD, College of Medicine, University of Vermont. He holds a PhD in chemistry from the University of, excuse me. He holds a PhD in chemistry from the University of Vermont and a BA in chemistry from St. Lawrence University. Dr. McKay has been recognized with numerous awards, including the President's Club Award and the Cannabis Scientist Power List. He holds several patents in extraction cannabis. Welcome, Dr. John McKay.
Thank you very much. I appreciate the opportunity to continue our discussion. I guess this is the part where I bring up the shared slides. I can do this twice in a row. That would be fabulous. Okay, done it. And I have to go to home. Let's see if I've done this correctly. There, I did it. Well, I do thank you for the opportunity to come back up again Friday the 13th, and also it's a time to be able to discuss more about extraction and sort of the episode 2. I'm not sure if it's a trilogy or it's a mini series, and I'm sure that somewhere along the way you can start to stream this. I need to get rid of some of the pictures over here, but I'll let that go. So continuing to talk more about principles and practices of assessing compliance manufacturing for cannabis. And there's so many things to unwrap. And what I'd like to do today is unwrap it a little bit more. This was a slide that we had from the beginning to know what we'd have learning by the end of the session. The first time we went through and we discussed about the principles and practices of extraction, looking at it versus concentration of compounds through separation, which is without solvent, and extraction, which is solvent. And now we're going to try and apply those a little bit more to what happens when you're looking at a facility that has an SOP, that has a lot of things in place that are check marks, but maybe underneath the surface, things aren't going as well as they look. And so we're really trying to get to identify the knowledge and then applying, wherever the gaps are in that knowledge base for you to do a great job being able to make sure that a place is compliant. I'd like to go back on one of the things I didn't cover in the first time, and that is for cannabis. And so cannabis has been categorized by humans, depending on the percentage of different compounds that are in the plant. Certainly it comes more from the fact of a lot of people still use the plant for euphoria. So if I was doing a word association, you would typically think, you'd say stop and say go. You say up, you say down, and people think cannabis and they think high. And that's not the entire case of this. As you look at this entire plant and the different types of seeds that go with it, with the genetics, back in the 1700s, 1600s, You had to grow hemp because it was used for clothing, and because on the ships it wouldn't wear out with the salt in the water. It was used for ropes, and so there were at one point in time laws that you had to grow cannabis to be able to make sure that you had enough ropes for the shipping industry and clothing. Um, it also has the ability of being used for the seeds, and the seeds have been always looked at as a very high protein profile seed of almost I won't say a perfect food, but certainly a very generous food missing one amino acid. I don't think it has as much lysine as we would typically need, but it does have all the amino acids that people could use as a food. It's still sold in stores for putting onto shakes and putting into salads and such for hemp hearts, which was without the hull. And then also you can buy hemp milk, you can buy hemp protein, you can buy hemp flour. All those different components are still coming from the same plant, though a different genetic makeup. And then of course you do still have the cannabinoids. And when we're looking at some of these manufacturing sites, we're kind of focused on that because that becomes something that is more amenable for what could be not optimal manufacturing and not following manufacturing processes. So we want to look at that a little bit more today. We had this picture from last time, and for those who didn't see the beginning of the mini series, this was the beginning of if you had a factory, you would have process parameters that you would be looking at. You would look at the facilities, you would look at the equipment, you would look at how it moved from one part of the factory to the other, which is your material flow from the time it comes into the plant to the time it's made into something and to the time it goes out as a shipment in a formulation. And then over on the far lower left, you're talking about documentations and records. You have to be able to make sure that you are following what you said you were going to do. So that is, stating what you will do and then following that and following the records that would show that you are compliant within that. One of the things that doesn't show up here is the fact that you want to be looking at the entire site. So the entire site of all four of those is the safety and regulations that guide the processes. If we take today and we make up this scenario that I will make up, and you assume that the site is compliant, it's got the specifications that are needed for wherever it is, material coming in, following the material to the record system, going through to make sure things are clean. And if you assume that the standard operating procedure, SOP, compliant with the type of manufacturing process they are using, that could be, it could be ethanol, it could be propane, butane, it could be a number of different soluble, as well as in fact it could be looking at just taking off the trichomes. Each one of those is a chosen manufacturing process, and you're assuming that they're compliant within the rules that you would have for the regulations of those. And to be fair, a lot of the things that you can do now for artificial intelligence or copying an SOP from a previous place or given to you by someone, it is possible to have a written procedure and not really understand what those procedures mean, because if you're writing it for a certain manufacturing, you're having committees that know what it's being used for, how it's being used, how it's being transported, how the equipment is being used, what manufacturer the equipment it had, what was the pricing, what's the service plan, what's the clean in place. There's so many things that would come up in a meeting. And then also you would have employees that are trained to be able to use those machines and different processes. That doesn't mean that that's always going to happen now. I've seen enough people who use AI and come up with terms that they bring to me, and I know that there's probably not a chance that they actually know what those terms mean. And so what can happen is you can have a place that has all of those documents, but also you're having the records and writing things down that if things move outside of that typical criteria, now you have a problem. I have a problem. I have a more recent problem that I would share. So I have a car and I have a Fiat, a 2012 Fiat, and the windows do not roll up. They don't roll up. But because I'm so clever and because I'm self-sufficient, I decided I would try and fix them myself. And I went to YouTube and I looked at the different ways of doing that. And I started to take apart the door and take off the window, which for people that know me, they already know, first of all, why do you have any type of screwdriver in your hand with your skill set? And so I did. I took it off and I said, this is really good. And I looked at it and it wasn't exactly what YouTube had told me. It was a little bit different on this model than the one they had shown me. And so now I have this window. And after about 2 months, of flailing at this, it's now at the car dealer with someone who knows what they're doing. So I know how to roll up a window and I can do it with power. I can press the button. I'm good at pressing the button. I know which button to press after the third or fourth time. And I know which way to put it and most of the time. But when it came to the fact that it was no longer working within the criteria that I thought Fiat was probably going to make that, it was a problem. And you would think that I would have learned from this. No, no, I did not learn from this. My washing machine broke. It stopped going round and round and round and round. It started to do back and forth. And I said to myself, I'm not going to go call up someone for $150 to come look at my $100 washing machine. So I took it apart. That washing machine is now out in the front driveway. And so from that, I know how to use a washing machine. I know how to turn the buttons most of the time. And I know how the water comes in and the water goes out. But when it doesn't do that, I came to a problem. And that's the problem that we want to make sure that we're not seeing within any manufacturing facility. But certainly as you're guiding yourself through the cannabis world of this is a new technology using new manufacturing, which sometimes the employees aren't coming from pharmaceutical or not coming from the food industry, then there can be that one little part in between that is doing the John McKay, I'm going to fix my automobile windows, which I don't, or I'm going to fix my very simple washing machine. And those things have to be able to be understood to know how they're doing that. And when I look at that, that's what we will discuss today, is what happens when something isn't working well. And there's people that will do that. And there's also manufacturing equipment where you have service people from different companies that are providing that. But sometimes those people aren't available. So let me go back a step from last week's or 2 weeks ago mini series on extraction. I want to go back because not everyone was at that mini series, and so we'll do one of those things that you see in the beginning of every single mini series. Let me go back to the beginning and do the highlights. Hemp workflow, extraction, isolation, purification. For me, it's a lot of different colored balls that are in that plant. And what you're trying to do is you're trying to eliminate some of the balls as you're going through a process to get to the balls that you want. So you're going through all these different colored balls, different types of filtration, different types of extraction, different types of separation, until you're down to the bottom where you have the compounds that you're looking for. If I look at that and I bring it into chromatography here showing you on the y-axis the height of different components that would be in a mixture. So if you have HPLC or any type of chromatography, you have a mixture and you separate them into their individual components, figure out where my hand is. And so now you have that capability, but as you're doing more and more purification, watch out for my fingers, which ones are coming up. And you have these two compounds at the last, and then finally you want this compound. So what you're doing is you're slowly but surely isolating those. And the thing that we did before is we talked about ping pong balls, iron filings, salt and pepper. And within those, you go through a sequence, a rational sequence of saying, okay, I'm going to get rid of the ping pong ball with a big filter. And now I'm left with the iron filings, the salt and the pepper. I can use a magnet. I can take out the iron filings. Now I'm just left with the salt and pepper. Pepper's not soluble in water to any extent, and so now you can take off the pepper and then you get rid of the water, and now you have the salt. So you have those sequences of what you're doing for an extraction based on chemical properties. There are two ways of doing that. So extraction with a solvent or different solvents, and you have separation, which is without a solvent. So a separation is a filter paper. When you're making coffee, for example, you have coffee grounds inside a filter paper. For example, you pour water through, the soluble compounds go through the a filter paper, but all the other compounds or the other grounds are a step behind. So that's a separation. The filter paper is a separation, and the water going through is an extraction where it's pulling out different components from that coffee. So you have those two pieces in place, but it's not always as simple as that. But when you're going back through, an extraction is a liquid or a liquid solid separation, extraction. So, for example, if you have oil and water, perfect example, everyone uses it. You have oil and water, and you have a layer in between the oil and the water. But all the oil up on top is all together, and all the compound and all the water molecules are together. It's only that one layer between the two that you have the separation, oil and vinegar, for example, the same thing. So you're doing that, and you see that one layer. Well, that one layer is saying, I'm not with you. I'm different from you. I'm going to separate myself from you. But anything that would be soluble in water versus soluble in oil would go into a water layer if you were to add it. So you have oil and water, you might add some other compound in there, and if it's water soluble, it will go to the water layer and not into the oil layer. So that's an extraction. You've taken something, you've added it to a mixture, and it's gone to the preferentially where it wants to be. Without solvent, it's mechanical. I'm grinding something, I'm making it smaller, I'm using a filter paper, I'm doing something that does not involve a liquid. I'm using size, for example, as part of my criteria, but I'm not extracting based on size. I just have a filter that has a different size hole. And when you bring them together, most of the time these are happening, I'll say simultaneously, but actually sequentially. And what happens is, now I've made up two words, so you can have an extraporation, where you have an extraction plus a separation at the same time, or you can have a separation, where you have a separation and you might have an extraction at the same time. I see Twee, so I know there's a question.
John, can you clarify? Because I know that you're talking about extraction and separation. And when you say that there's no liquid involved, when we're talking about very specifically bubble hash, water is a liquid. And my understanding, and please correct me if I'm wrong, is because um, what you're targeting is not soluble or even it's not soluble in water, that would still be considered a separation, a mechanical separation, correct?
That's a
Doesn't
mechanical
dissolve in.
separation when you're looking for the trichomes and you're looking for the cannabinoids, knowing that within that your criteria is that you're looking for those components. And so they're not soluble in water. So
Correct.
if I'm, for example, if I'm washing off grapes, I'm not assuming the grapes are going to be soluble in the water, but I am taking off the pesticides, for example,
Mhm.
if they're on there, or anything else that would be on there. So I'm wanting the grapes. In this case, they're wanting to have the trichomes for the bubble hash. There are other compounds that would be soluble in the water, but it's not based on the criteria of what you're after. So that would definitely be a separation. It's just
Okay.
that you're getting what you want, but the other ones are still behind in the water. So we'd also gone through best. And whenever you're doing an extraction from natural products, for example, I speak of it, botanical materials integrity maintained to assure the concentration of the desired compounds in a natural form. And just what Twee was speaking about, you're wanting to have the trichomes in their natural state, not broken up and not being, outside of the water, so not part of an extraction. At the same time, you're wanting the other components to go. So you're looking for desired components in their natural form of what you want. And then E is effective concentration with efficacy, efficiency, economics in predefined boundaries. So even the first two I'm talking about, what I'm looking for. I'm looking for desired components, and I'm looking for predefined boundaries. Safety is where we're really looking towards everything in that situation process, from the workers to the plants to the final product is being made that's used in a formulation. And then critically is testing with modern technology so that you're looking at every facet of that. You cannot manage what you cannot or will not measure. You need to have measurements. And we'll speak about that as we look through my hypothetical situational process. If I move towards that, and something we've already spoken about is coffee, and we've spoken about this one last time, that as you're looking between a regular iced coffee and a cold brew coffee, regular iced coffee is made through the um, through the process of making hot coffee and then putting ice into it. The other one is cold brew coffee, where you're really starting off with cold water and brewing it for a very long time because the extraction process is there. So I happen to be in as always, I'm in some sort of hotel or something, but I'm in a Marriott property, and this morning they'd run out of coffee, so I had to take some cold brew. And the cold brew definitely tastes different from my hot brew and from my concentration. So both of those have a different flavor. And the reason that is because less compounds are in a cold brew because it's measured, it's used with a ice water in the beginning. And so you have 1,000 compounds, where a regular hot coffee has 1,500 compounds, but then you make it cold. Those are two different products, even though your end game is something that is cold and is a coffee. Very different products. If I look at what we're doing, going back to the cartoon, and that is discussing everything that's happening within an extraction process, within a manufacturing process, within a formulation process, within something that's making products that go out to the market, there's a lot of things to look at. And we want to examine something with this situation I have contrived of. So each one of these is important for safety. Each one of these demonstrates a respect for whatever manufacturing process it has. And each of these demonstrates a natural product and its role in the world. So we're going to look at those. And the key thing is looking at process parameters. And I did this in the last scenario, and that is, what's the depth of a knowledge and experience? So through questioning and also understanding that these questions are important for finding out not only is the process there, but what happens if something goes wrong. And so what's being controlled, and how do you know the control? Is it temperature? How do you know the temperature is being controlled? Are there limits that are selected over others? Is there evidence that the process is in control? Is there some sort of read back where I have, these are my standards, this is my internal standard, this is my regulatory standard. And so you have different standards in being able to make sure that your temperature is within a certain range, or your concentration is a certain range, or your yield is a certain range. And what happens when they're not in control? That tells you what's understood versus what's just being read. Is it something that just came off II? And part of any process, as you well know, is the training. It turns out that there's humans involved. And knowing that they can be able to talk about cause and effect, not just recite points. So those are the key parts that you're looking for, because it'll bring through, do they really know, as does the entire manufacturing facility know? If I look at this slide with you, and that's examines of common solvent-based equipment and parameters, and I just picked three. There's hundreds of ways of doing it. I've just picked three. So I've got carbon dioxide, I've got ethanol, I've got propane, butane, and each one of these has a different component in those that make the instrument viable, that passes inspection, that the fire department looks at, that the engineers look at, so that that you have different components that are being used that are very different from each one. And then the control focus is what am I controlling based on these that allow me to know that I'm having a compliant product and it's meeting the standards that I have. So each one of these talks about, for example, CO2, you have pressure and temperature tuning, you have solvent recovery, you have many factors involved. In ethanol, you have temperature of ethanol, you have the flow rate of ethanol, you have how many times you're using it, how many the different components that you would use for crystallization. And with butane, you have your VOC, you have closed loop. You're making sure that things are not there's no electronics anywhere near it, and so you have all these different criteria that you have for those versus CO2. No good, no bad. They're just different. You also have the solventless, which would be a separation. So you have water hash, you have rosin, you have dry sift. Each one of these is a different parameter also where you have different types of compounds. The water hash, for example, having the water that you have, really cold water, or you have water that's being able to be moved back and forth, which brings me up to my nightmare of washing machines. But within that agitation, the gentle agitation, each one of these has a different parameter as well as what kind of ice is it? How was it made? Because you can have, really hard ice, or you can have, slushies. Each one of those is important. And the trichome purity itself. So the trichome is a little tiny bubble of a material. And we'll look at that in another slide. But you're knocking those off. So in Vermont, when we have, I grew up in Vermont, just show that I'm unique. But and that is when I'm going through it when it's time in the late fall, early fall, late summer, early fall, and it's time to pick the apples. You don't hear the chainsaws coming out to cut down the apple trees. You actually pick the apples off, and so that's much like the trichomes in many respects. I see Twee is there, so I will stop for a question.
Yeah, no, just a quick note, for butane, and
Yes,
that would be as well for propane, just for
yes,
clarification,
yes.
with the attendees, because generally you'll see butane and propane, um, in one extraction.
Yeah,
Sometimes it's
sure.
just strictly butane, sometimes it's strictly propane. If they have equipment that is rated for the PSI for, I believe propane has a higher PSI. Is that correct? If I can
No
recall.
idea. I'd like to
Okay.
say I know, but I don't.
Uh, I'm going to take a wild shot. I think it is, but if you have a piece of equipment that's rated for the higher PSI, then you can use both. But if you strictly have a lower PSI piece of equipment and it hasn't been rated for that specific LPG gas, then you cannot use both. So just quick point of clarification.
What's LPT?
Uh, liquid,
LPP?
liquid petroleum gas, right?
Yes. Thank you.
You're testing
Good.
me. You're
No, no,
testing
I'm
me.
not testing. No, 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 no. I wouldn't do that. Not twice, anyhow. All right. So now we have each one of those. So each one of these has a different ability, whether it's solvent or solventless. And we went back through, and there's different this has supercritical CO2, but it's the same is true with ethanol or anything else, and that is things go very quickly in the beginning and then they start to slow down. So you have mass transfer, you have thermodynamics happening. So all of these have real science behind them, but that doesn't mean that you have to have real science doing it. So, for example, I'm sure there's real science that's bought into washing machines, which I am not totally familiar with anymore. I know how to break one. If you need one broken, please call me. You have the email. I can get there. So different types of trichomes on a cannabis plant, and the ones that most people are looking at are the captate trichomes, which are the lower picture that looks like a ball with the thing sticking through it. But when you look at that, that's mostly what they're looking for. And there's different sizes of those depending on where they are on the plant. But you also have the other types of trichomes, and they're probably not coming off with the water hash, so you're not getting everything off. But you really are trying to get them and sieve them by different sizes, and you're also trying not to get off chlorophyll. There's a lot of things that go very well, and with the real scientists going through and making sure that they're getting the cleanest amount of trichomes. But even there, you're dependent on the fact that you don't have pesticides in there at the same time. You don't have other things that you're also trying not to get. And so each one, it's true of all types of extraction and separation. But it goes down to understanding what's in there. And you can do something very simple, whether you can see over on the left-hand side, bulbous red material, which shows that I still can't do a thin layer chromatography very well. Some people would have one much nicer, like over on the right-hand side. But I can do chromatography. And in the middle, we have a number of different standards that are in there. And you can tell through that how much material you have in each part of your process. So from the starting material, what is it there? In the spent material, what's left over? In the material that you've collected, how much is there? And so you have all those different components, as well as what you're doing for more and more on the purification, as we saw earlier in the slide with all my different colored balls, and then the chromatograms on the right-hand side.
So John, can you explain what a chemovar is for those who don't know?
So chemovar is, oh, did I write it somewhere? And so many people speak about strains of cannabis. You can't have a strain that's bacteria. That's a whole different thing, but it's gained so much popularity that people still talk about strains. A chemovar is something that you can come up with a word that's a real variant, a real genetic plant, but because cannabis has been is not federally legal, you can't really, try and patent something that is a illegal compound or type of genetic. So I can have Rome apples, I can have Washington crisp, I can have sugar crisp, I can have all the different types of apples, and that's a different variety. But and it's a registered variety that says this tells me I have a Washington crisp apple. I've grown this apple. You can't do that with cannabis because it's federally illegal. So you can't have a registered variety of an illegal plant.
Very good, thank you. And then also just for a quick point of clarification, when we were talking about the PSI for butane and propane, I just looked it up really quickly. So PSI is pounds per square inch,
Yes.
because sometimes we speak in acronyms and assume that everybody understands. So I'm trying to make sure that everybody understands
Yes.
any sort of terminology that we're using. And the PSI for propane is between 105 to 145 PSI, and butane is between 15 and 30 PSI.
So which one's higher?
Propane.
Just testing.
Yeah.
Yeah. So when you have a car and you have a PSI, so I haven't broken my tire yet, and I probably won't repair it, but a PSI, you're between 32 PSI of what you put into a tire, and some of them are higher, which is the same rating that you have for a soda. It's about the same rating. Free information. We talked about solubility and we talked about the oil and the vinegar. This would be the same part where you have a real octanol versus water. There's a ratio between the two, and where a compound goes when you place it into that mixture, how much goes into the water, how much goes into the octanol. You have that rate, and it's called log P. And so you have that distribution. And so based on that distribution, there's other compounds that would be like that. So when people talk about like dissolves like, if it's something that's water, it's going to be liking water. It's going to stay in that layer. So they have that as far as all of our chemistry talk. But also you see it in every single day of things that go into different compounds, different solvents that you're trying to get into. If I look at that with CO2, carbon dioxide is dependent on its temperature and pressure. And so the greater the pressure you have, the more soluble it is, the closer the compounds are together, the closer you have for polarity. And so each one of those is going to be a contributor, but each, as we see on these two graphs, I have the one on the left-hand side. If I talk Fahrenheit, because I'm in the United States, I got 107 degrees Fahrenheit. The one on the right is 127 degrees Fahrenheit. And you can see that the lines are different as the pressure goes up. So on my y-axis is solubility, how much is soluble at that temperature and pressure, and across the bottom you have pressure, so increasing pressure. If I look at the one on the left and I see the CBD and CBN, two compounds that are typically out in the market now for sleep and one for anti-inflammatory, you can see that over on the right-hand side, they've switched. The CBN is far more soluble than the CBD, even at the, just 20 degrees difference in temperature. The pressures are still the same, but you can see that a significant difference between those two molecules as there's a difference in pressure and temperature. That would affect it if you had something that wasn't being monitored as far as temperature, you're going to get a far different result of that product based on the fact that you're not monitoring the temperature specifically for that extraction. So you will have a difference in what you're getting for a product. You'll have it either more or less than what you anticipating.
So, John, for those who aren't familiar with CO2 extractions, you can control the temperature and the pressure of how you're running your extraction. And so, based off of that, and based off of the chemovar that you are extracting, that's how you're going to adjust your extraction so that you can try to direct that extraction towards that very specific cannabinoid. So hemp versus, let's just say, cannabis. Cannabis has a higher THC content and you want to extract THC. But if you have a hemp product, which is primarily CBD, you may extract it at a different temperature and pressure, as well as any strains that may be higher in potency for CBG or even CBN. So that's just a quick, down and dirty for those who aren't familiar with CO2 extractions. And John, you can if you'd like, you can elaborate on, from my understanding, for supercritical CO2, there's a wide range of temperature and pressure that you can run the extraction at, and it's still a supercritical CO2 fluid, correct?
Correct. So there's different phases that happen. So we know CO2 is in the atmosphere. I'm breathing it now, and then we have CO2 that's a liquid, and it's under pressure. Most of the time, we're not seeing that, but we do see dry ice. That's CO2 in a solid form, and we see that many times it doesn't stop at liquid. It goes right into a gas, so it sublimates. It goes directly from a solid to a gas. And then you have supercritical. And supercritical goes to a certain temperature and pressure, which are certainly not in natural for this planet. And so you have to put it into a pressure container, build up the pressure by decreasing the density and also increasing the temperature. And so now what will happen is, if you see it on YouTube, you will see it go from a gas where you can see it through a container that has glass in between. Well, not really glass, but something that's transparent. And therefore you can see it go from a gas, then you'll see it go to a liquid as you raise the temperature and keeping the volume the same. And you'll see it turn into a liquid. And then as you increase the temperature further, the liquid will turn into a dense fog type of thing. And that's why early on in the early 60s, late 50s, it was called dense fluid. It wasn't called supercritical fluid. It's called dense gas because it was dense. And so the CO2 at supercritical acts like a liquid and also acts like a gas. So it will go into things like a gas, but it will extract like a liquid.
And because of this characteristic, Again, dependent upon what they're extracting and the extractor themselves and the finished product, what type of finished product that they're aiming for, they may choose to extract with CO2 versus an ethanol or a butane, propane, water. So, okay, thank you.
Yes. So I go back to this slide. On each one, the middle one is really focused on safety and risk management. And then I put the other slides around the outside because I can do that in PowerPoint and I think myself clever. And so over in the top left-hand corner, I talk about the process. The top right-hand corner, I have the instrumentation. And then the bottom right, I talk about where the flow is. And then on the bottom left is the documentation. But in the center of all this, for all of these things, the reason we have it is for looking for safety and risk management. And that's what we'll make up a story about now. This is supercritical fluid, typical, a system. Over on the left-hand side, lower left-hand side, we have the container with the CO2. We have different heat exchangers to be able to make sure it stays in its liquid form. We have ability of adding other solvents if we wanted to, ethanol, ethyl acetate, hexane. Any of these other things could be added in. And the key is that these two in the middle that I'm hoping an arrow shows up besides just my computer, and that is, this is the extraction vessel. So if I have an extraction vessel, I load this vessel here, which could be about 1.5 meters tall. And within that very sealed wall of a special manufacturing, I place the plant material into here or whatever it is. We'll say it's plant material. The plant material, the CO2 will come up in this case from the bottom, exit out the top. It then goes through and can be collected in other vessels. Some have 3, some have 1. But as it goes through this process as a supercritical, it can also come down here. When it reaches this vessel here, the pressure is released. The CO2 it becomes a gas, the material is left, and then you collect it on the bottom. So the criteria is CO2, in this case liquid CO2, coming through, going up through an extraction vessel that's been filled with a botanical, then over here being able to be collected in one of these 3. When I look at this, because you have different types of material that can be extracted, in many cases the flavors and fragrances, are typically the terpenes, very volatile, so those can come off first. You'd be able to collect those also in a vessel. Then you can also collect out the cannabinoids. But the key is that because it's sitting behind this very solid piece of steel, you don't know what the actual extraction efficiency has been, and that's the problem that we'll try and solve today.
So John, thanks for touching upon extracting more volatile compounds such as terpenes. And so when you're conducting a CO2 extraction, and I would say it's the same for butane and propane, there are valves on the extraction equipment where you can close it off. And so you can literally do a CO2 extraction, and knowing that terpenes are more volatile than cannabinoids, we can close off the valve and we can extract the terpenes and actually remove it and change the collection vessel so that we are separating it from not only the cannabinoids, but also if you're not doing, and I'm going to say a whole plant extraction, whole plant meaning it's got everything. It's going to have the terpenes, it's going to have the cannabinoids, it's going to have the waxes and lipids and the heavier compounds that will be extracted later. That might be considered a whole plant extraction, whereas there are extractions where they will, close off the valve and then collect exactly what they're looking for so that they can separate it a little bit easier. And it makes it easier when you are further processing it for refinement or purification. And then also I wanted to ask you very specifically, what sort of safety risks do we need to consider when you're doing a CO2 extraction, but it has a cold solvent like ethanol?
Each one of these components has to be um, for the solvent that they're using. So specific valves, specific materials of construction, the correct seal material, the correct rupture disc. So everything that's being used. So when you're doing a co-solvent with ethanol, that system has been made compliant to be able to mix those two together. So by definition, the manufacturer would have already gone through all of the engineering tests and have gone through everything compliant with that. So when an inspector comes in, they make sure that they see a system of record, that it meets the same serial number that the manufacturer made, and it had exactly the same components that the manufacturer had gone in and started up through their qualified service engineers. If you have something you've made yourself, then it has to go through engineering also. So there's firms out there, especially for any type of industry, but certainly the cannabis industry, to be able to say this meets the engineering specifications for materials of construction, the valving, and the safety that would be involved.
And we will be diving deeper into uh, like engineer approved equipment as well as extractions on May 8th. So make sure that you join us for the May 8th webinar. We're going to speak very specifically about how Denver, the PE for Denver actually created the codes and standards for cannabis extraction operations as well as botanical operations. So make sure that you join us for May 8th. So back to the co-solvent, just for my clarification, does it generally get introduced into the vessel, the extraction vessel, along with CO2, or is it before or is it after?
It's, can you see my arrow? Do you see a little arrow? A little, my
I
mouse?
think so. Yeah.
Okay, this is the mixer. So the pumping would be separate from the co-solvent in this case and the CO2. So they come into mixing before they go into the extraction vessel.
Okay. Very good. And how does that work? A
Really
liquid and
well.
a supercritical fluid, like it mixes really well.
It mixes
And
really
does
well.
it form a liquid then? I would suspect.
It forms so when it's done well within the compliance of what the percentages are, it will be a single mixture. It will be
It would be
supercritical.
considered a liquid, correct?
No, it doesn't have to be considered liquid because I also have a heat exchanger right here.
Okay.
So I can bring them both into a gaseous state.
I see. Very good. Thank you.
Welcome. So these are as Twee was mentioning, these are the types of pressures that you would be mounting to make sure you had, and that you have rupture discs, you have the right materials of construction for valves, for seals, for everything that's within that process. So, 6,000 PSI, 25 degrees C centigrade. If I look at that, when I have the ability of at that pressure and temperature, that CO2 just on its own is equal to ethanol. So it has nothing. So the CO2 is certainly tunable, and you can have something that emulates the same um, extraction proficiency as ethanol. But and then you have different, as we had mentioned, you have different products that you can collect based on their solubility and when you release them from the gas. So those things all come into components. So you can have different types of compounds. Way over on the right-hand side, it might be terpenes with a lot more terpene power. Other things that you're trying to collect based on the pressure and temperature that you release the CO2 from. If we look at this beginning of the scenario, I hope that the movie plays. Maybe it will, maybe it won't. But as I'm looking at the left-hand side, that would be the extraction vessel, and I'll be putting a container into it. that was some sort of excitement. I know you probably want to watch it again, and maybe on YouTube it would be. If you, what you're doing is you're filling the vessel with the material and then placing the vessel into the tube. So many times what's happened in the cannabis industry is people have been putting plant material directly into the vessel because they thought they could put in more. This is actually the better way to do it, is to have a container that goes inside that, so that you're taking the container outside of the room, being able to fill it, then bringing it into the room, extracting it. The one on the right-hand side is after an extraction has been done, so that you can push it out like a Tootsie Roll pop. I should mention a product. You can put it out as a roll. Those are given many years ago by Proverty Labs out of Milford, Massachusetts, and doing some of the work that they were doing early on there. And you can see that now you have this entire hull of material. And that's what we're going to look at, is because we're going to look at what happens if you don't have a full extraction, and how you're able to monitor that material and be able to make it so that you are extracting everything. When I also look at the collection side, this is open from the collection side with a little bit of ethanol and gas. So what happens when you're doing the collection is spinning in a cyclone type of wave. It gives it a lot of time for it to be able to fall out of solution, and that's how you collect it. I'm looking at that great big piece of cylinder material that I showed you before, and I show it on the lower side that if it was standing up, that's what it would look like, but I turned it on its side, and so now you see it from the bottom on the left-hand side to the top over on the right-hand side. And we'll examine this a little bit more over the next few slides. If I did something really well, and if I was taking at least 3 samples, and I show 3 samples in the middle there, with a sample from the bottom, sample in the middle, sample from the top, then if that was the case, and if I had extracted it correctly, then I would have less than 1% on any of those sections. Because as we saw before, there's a diminishing return, there's different things that's going to happen. You're only going to grab out, 90% of that, 95% of it. So over on the right-hand side, I show a chart where you can see a color of how much was left in there. So this is an acceptable extraction. You've collected it all. You've tested 3 different layers. And so you know that you've got everything out. It turns out that that isn't always what happens. My windows go up and down, and then once in a while, they don't. So if I assume in the case that my particle size is the same, we'll call it 8 millimeters. I'm going to assume that I packed it with the correct density. I didn't, not too hard on some parts, not too weak on others, so my density is equal. I assume that the carbon dioxide coming up from the bottom is pure every single time. I assume that the top of the vessel is closed, it reaches its temperature, it reaches its pressure, and then it opens up. I assume that it's reached these conditions and now has moved through the process for being collected. And I assume that everything is soluble. I assume that everything is clean. I assume that I have set those conditions correctly. And yet my windows don't go up or down. And now you have to break down the spent material. It's not raffinate, it's spent material. It's exhausted material. When you do an extraction, for terms, and you wash your clothes, the material is being selected through the water and through the soap. That's the extracted material. The raffinate is the clothes. I'm assuming that you're doing a washing machine, well, when yours works, that you're doing that washing machine and you're keeping the clothes. In the cannabis world, we're not keeping the material. That is spent material. That's exhausted material. And so that's a difference, another, phrase and phase. So I'm going to look at this thing, and I see up on here, I see these clumps. I call them moon rocks. That's definitely not a scientific word. But what's happened is that material has gotten some cannabis oil around the outside, and it has now made it almost impenetrable for the CO2 because the CO2 is going around it like a beaver dam. It's going around it. It's going around it. It's being clogged. And so nothing is being separated inside that material. So it's got a little bit of an oil layer on the outside of it. I call it a moon rock, but if you open it up, that's going to have a very different um, amount of cannabis. So that's one problem I can see. I see the other problem is it's only progressed to this point. It's only gotten to this point, and I can see that it's kind of green, light green in the middle and kind of a little dark here along the sides. That tells me something's that it's not uniform. I look down here and I see, lighter and lighter green, but I also see these other parts that are dark in the middle. And I look at that and I say, okay, this is not an extraction that has gone to completeness. Why not? If the conditions were the same and it was packed the same and everything else, then why not? And so now you have the ability of taking analytical data because you can't manage what you cannot or refuse to measure. And so now you've got this problem. And if I look at this, I took another cut of this. I cut this here and I look on down the side. So this is the cylinder down, and you can see there's dark green towards the outside and lighter in the middle. This tells me that something has happened such that the CO2 is now moving up the middle, not to the outside. It hasn't been dispersed throughout the sample. It's now channeled like the Colorado River, like the Mississippi River. The Mississippi River does spill out at times, but most of the time when you have a river, it's staying in those conditions. And that's what happens when CO2 or any other solvent is now found a path that it's going to keep going down and it's not going to go to the other parts. It's found a path of least resistance. All of those come into play, which means there's something that has to be done. Either it's a part that's always going to be there that you're always seeing, or it's a part that you're being able to know that something else has to be changing. And in this case, the CO2 was going too fast. They had too fast a flow rate. They also had not packed it uniformly. So the carefulness of packing, so they packed it a little too heavy on the outside and not hard enough in the middle. So it's hard to do that. But the other part is when you did the analysis, let's look at, so first of all, good is not a number. Bad is not a number. It's not a number. That looks good or looks bad. Neither of those are numbers. You need numbers. And so in this case, what I've done is I've measured 1, 2, 3, 4, 5, 6, 7, 8, 9 different parts of this problem. And you can see that within that, I have different percentages. There's a lot more material out here. The beginning material was less than 10, so actually you have a greater amount up towards the front because it hadn't reached the end. It hadn't gotten out the tunnel. But on the bottom, it looks like I got that. Remember, that was the light green. Remember that we saw one section on one side that looked darker than the other side. And indeed, that is the case. You're seeing that it's lighter over here, but it's still too dark. And so each one of these allows me to take something that is a visual into something that's data so that I can look at it and say, what do I have to do? In this case, it needed to do a static and dynamic flow. Static means I'm making a cup of tea, I'm soaking it. It doesn't matter if it's CO2, it doesn't matter if it's ethanol, it doesn't matter if it's propane, butane, or insane. It doesn't matter what it is, it's being able to know that it didn't come out. So I soak it, that's called static. Dynamic means I flow. And so that's what we were able to do to solve this individual challenge. Tweet.
And while you're looking at this color key, that is specific to the percentage of cannabinoids that are found in the material at each level. So
Correct.
just to be very clear, that's what that
Yes.
is.
Yes. So I go back to the cartoon. Process parameters. you can have an SOP that looks great, but you also have to know when things are not falling into where you thought they were, or you have a process that historically every time is going through that because the process is incorrect. In this case, the process was incorrect. They thought they were getting lots of material, but they were nowhere near the optimal rate of what they should have been getting. The facilities and equipment. Do you have the right equipment? And as Twee had mentioned, do you have the materials of construction? Do you have the filters? Do you have the rupture discs? Do you have things in place, certainly with propane or butane, that you have all those things, but you don't have any electrical near them? You have everything that's set up same way with ethanol. Do you have the right temperature of ethanol? How long is it being used? And then you have the materials of flow, where they move through the factory. It'd be best not to be grinding right next to your extraction equipment. You have a different room for that. You bring it into the next room. You do an extraction. You bring it into the next room for some people doing winterization. You bring it into the next room for further purification, or you're doing evaporation of some sort. You're bringing each one of those into a place that is material controls, that has the facilities and equipment and the process parameters so that they're for whatever process that is. And then you have the documentation records. What did I do for each run? How much did I put in? How much did I get out? What other things came out at the same time? Because those are important. And everything comes out to the great big huge thing, and that's the safety and the regulations. So each one of these parts is critical. That's what we discussed today, going back through with this criteria, but something that happened to be a CO2 extraction. But the same thing is happening no matter what solvent it is. You're being able to do the same thing. You can still go through and know each part. You can do the same thing with separations, with hash or anything else, because you're knowing what the size is and you're weighing the size based on that. When does chlorophyll come out? When does something happen where it's out of compliance? Each one of those is critical, but each one of those is critical to the safety and how things are done. And with that, I ended. If there's any questions, um, happy to answer them. Thank you so much for your time. Thank you for episode 3 of the mini series.
We
Episode
don't,
2 of the mini series, wouldn't that? Yeah, episode 2.
yeah, we don't have any open questions right now. So if you have any questions, please put it in the Q&A chat. So I know that you discussed in detail CO2 extractions, and I know part 3 is going to discuss some of the other solvents. So definitely make sure that we join for part 3, which is going to be on March 27th. So when we're looking at PSI and temperatures, just a quick recap, CO2 is going to have the highest PSI and also the highest temperature. It's generally a warm or even a hot extraction. And then propane, which is a higher PSI than butane, and also a lower temperature for extraction. And then butane, which has a lower PSI and, still a cold temperature for extraction, but not as cold as propane. So, if we're talking about gases or supercritical fluids. And then when we start talking about ethanol or other solvents, let's just say for instance, ethanol, you can talk about like a cold ethanol extraction or even a hot ethanol extraction. So it all depends on how you're extracting. And then for those who maybe missed the first part of your series, your first series back in I guess like 2 weeks ago. So can you explain the difference between extraction and extraporation versus separation and separation? So just so
So I
that
made
we
up
make
these
sure that
words so
we're very
that
clear.
they don't often roll off people's tongues. They roll off mine, and even that's only 50% of the time. So extraction truly is a liquid or liquid solid. So I'm taking from a liquid coffee, and I have a coffee bean that's a solid. So I'm taking a liquid and I'm passing it through a solid and taking off other liquids into a uh, solvent that it's happy with. If I look at a separation truly does not involve a solvent. You can have a carrier, like we spoke about with water. You can have a carrier, but it's not being part of the active part of the extraction. So water can be a carrier. For example, I would say one of my former managers, if anyone could manage me, is even a dead fish in a fast moving stream looks like it's making progress, but it's not being extracted. It's just flowing. A leaf is flowing, which is a far better word picture, perhaps. And so if I'm doing that, and then my leaf gets caught in a beaver dam, that's being held back. Okay, so I have both of those in place. When I made up separation, then I knew that I was saying that my primary part was I was filtering, and so therefore I was, holding back. But at the same time, I was extracting. So I had two things happening simultaneously or so closely sequential that it looked like it was simultaneous. And so you have that. I have extraporation where I really believe that I'm doing majorly extraction, but I do still have a separation component of it. So those are not words that are in any other's dictionary besides the John Mackay word dictionary. So they are truly made up. But
Okay.
those are the definitions of them.
So under John Mackay's dictionary, extraction and extraporation is one and the same. They're synonymous. And
No,
then
extraporation, they're not, because you have a
they're
separate component.
okay.
So if I was
I
doing a pure extraction, then I would have vinegar and oil, and I would add another component, and then that would go into to one of the solvents or the other. That's an extraction.
okay. And then also the same separation and separation, not synonymous.
No,
And then again, just tell me the difference between separation and separation, if I'm saying
yeah,
that
that's
correctly.
that's that's going into my brain. There's very few people that come out well. I don't pay for counseling after any of these sessions. So if I go back through and I was making coffee, then for me, I would call that extraporation because I'm really trying to I'm really trying to gain the coffee, the liquid that's coming down the bottom. I want that, and I'm holding the other one back. A extraporation perhaps is one where I know that I'm holding it back, so I could do that even with winterization, and so I would be doing a separation based on solubility, but then I would pour out the other material, and I would be saving that and holding the waxes back. So it's a fine line, and probably more in my brain.
Okay, very good. Um, So you were talking about very early on, you were talking about how specs should be listed in that, or, the SOPs very specifically. Do they know, um, what the SOPs are? Can they use AI or even, borrow or even buy a set of SOPs to submit to the regulatory authority for approval? So what, I've noticed when I do inspections is when you're walking in for an inspection, unless you're reviewing the SOPs that were submitted to the regulatory authority, and it could be either one and the same or two different ones, just depending upon which state or municipality that you're working in, the specs should always be listed. So your critical limits, right? Your action levels. In addition, what are your acceptable operating parameters? Because that could be very different than what would be considered, say, an action level or even what's compliant. So definitely need to make sure that you're reviewing the SOPs and seeing kind of how the operator is listing that. And what I have recommended to operators is I would say, well, make sure that your logs also state that. Because if I come in and I ask your employee, what the critical limits are, what are your operating limits, and they don't know, that's obviously lack of demonstration of knowledge, right?
Yes.
So, I always highly encourage them to do that, to make it very easy. Because a lot of times when they're working, they're just working. They don't have the SOPs right there in front of them. They just have their logs and they've been trained on the SOPs. So, I would definitely make sure that when you're doing an audit, looking at logs and SOPs, that you review that. And then the other thing that you had mentioned, with respect to, the difference between, say, cannabis extraction operations and, like food facilities. I just want to make very clear that retail food, and for those of you who have worked in retail food, very different than manufactured foods, different set of regulations. If you think about it, retail food is generally ready to eat, or, and eating very soon, either right then and there or within a few hours, unless you refrigerate it, but you don't have the extended shelf life that you would for manufacturing. Right. So manufactured foods or even dietary supplements, that's an extended shelf life, which, when you're talking about that, you have to make sure that there's testing for shelf life stability. And, I guess we can get into that in another webinar. But I just wanted to make that very clear, because a lot of times you may assume that just because someone has food experience, food establishment experience, it's not the same as manufactured foods. Very, very different.
Where does
So,
Oreos fit? Because well, not Oreos, any cookie with a cream fill in the middle.
well, that's manufactured food, right? Unless they're making it fresh right then and there in the bakery. And it's not an Oreo, but it's like an Oreo,
Yeah, I can't
right?
guess.
Because
Yeah.
that baked good is probably not going to contain all the preservatives to ensure the best by date or best quality. Um, it's not going to be good, 30, 60, 90, or, several years later, right?
Well, that's
So,
why I usually go to the stores that have them several years later. They're cheaper.
yeah, yeah. So I just wanted to make some of those comments. So I still don't see any questions in the Q&A chat. So we will probably just end this early. So
Thank you.
please, is there anything else? Last thoughts for part 2, John?
No, I'm quite happy with what we talked about and being able to bring it full circle. So
Okay.
thank you for
So
attending, and thank you for watching the if they're streaming, because now everyone else afterwards will stream this. So
okay, so Amy, can you from the slide to promote the next webinar? Where's the link? Oh, where do we stream? Okay, so you can stream it on the AFTO YouTube page, and AFTO generally will have it up on the AFTO YouTube page within 1 to 2 weeks. They've been really good about posting it, and if you registered for the event, you will generally get an email stating that it's up. But if you just subscribe, you'll see that, and you can see all the previous presentations. So this is episode 3. Episode 4 is going to be part 3 of the trilogy, also with John McKay. And I hope you guys caught all of his little jokes. He's a jokester in the presentation. So that will be on March 27th between 3 and 4:30. So please join us for that. But if you can't join us, just pop on into the AFTO YouTube page and you can review the information. And if we're talking about things that you need to digest a little bit more, then, you can rewatch it as many times as you'd like. I will also say this, for those of you who have specific questions that maybe you kind of figure it out after the presentation, after the live webinar, feel free to shoot us an email. We can definitely address it during the live webinar, so that if you need further points of clarification, we can go ahead and do that. Okay, very good. Next slide, please, Amy. So if you would like to join us for the AFTO Cannabis Hemp and Natural Psychedelics Committee, please join us. But Jonathan Gerhardt, who is the bureau chief for the Cannabis and Hemp Bureau with the New Mexico Environmental Department. And then myself, we are the co-chairs for the committee. And like I said, if you would like to join us, next slide, please. You can go ahead and scan the QR code or just go to the AFTO website and you can join there. All of this information is free. You don't have to be a member. However, we really encourage you to be a member because there's other information that you can access as a member. And we also have our annual education conference coming up in June. So if you'd like to attend that, we do encourage you to register for that as well. And so I'm going to take another quick look really quickly to see if there are any questions, and I don't see any questions. So yes, so please join us on March 27th. Thank you, everyone.
Thank you.